Okay, we should probably get started. Good evening, everybody. Welcome to day 3 of Brain School 2019. More amazing brain things to learn tonight. It's great to see so many of you back during spring break week, so thanks for coming. Somebody was saying before they were thanking us for doing this, which we greatly appreciate, but in response I said, well, thank you. We're having probably more fun than you are. We really enjoy the opportunity to share some of this stuff with our neighbors, friends, and colleagues, so thanks for being here. So it is my pleasure tonight to introduce the topic of traumatic brain injury, but before I do, I want to remind you of the other stuff that's going on. So one of our students, Jesse Mann, was just down in the lobby putting on a demonstration that I think several of you may have seen. And then immediately after the presentations again this evening, Dr. Fox, who you heard last night, will be out in the hall with his friendly brains again. For those of you who either haven't yet had a chance to handle a human brain or already have and are so compelled you want to do it again, you may. But please try to give an opportunity to those that haven't had a chance first. And then tomorrow, the final day of Brain School 2019, we'll have a group here from the Alzheimer's Association that will be providing materials about Alzheimer's disease before the talk, during the 5 o'clock reception, basically. And then at the conclusion of tomorrow's program will be the graduation ceremony for all of you who have made it through this, who get your certificates to go out and do things with brains. So. All right, without further ado, it is indeed my great pleasure this evening to introduce the speakers. So as I said, the topic for tonight is something that I think everybody is aware of and interested in, that is traumatic brain injury. And once again, we're going to hear from a scientist, a researcher, and a physician who both work in this area, and I think you'll find it fascinating. So the two speakers this evening will be Dr. Pam VandeVord, who is a professor and serving as the interim chair of the Department of Biomedical Engineering and Mechanics at Virginia Tech, who does research in the area of traumatic brain injury. And then Dr. Melanie Prusakowski, who's a physician at Virginia Tech Carilion School of Medicine at Carilion Clinic in the emergency department in pediatrics, who deals with treating patients with traumatic brain injury. So between the two of them, I think you'll get a very nice coverage and picture of this very exciting topic. So without further ado, I'd like to welcome Dr. VandeVord to give the first presentation. Pam.
Thank you, Mike. Can everybody hear me in the back? Okay, great. I'm really excited to be here for Brain Week. This is my first time that I've participated, and I've realized through all the emails that I've been getting, this is going on across the whole country. This is pretty exciting to be here. Is this gonna come on? Okay. All right, so as Mike has mentioned, I am an academic researcher. My name is Pam VandeVord, and I work in traumatic brain injury research. And tonight I'm going to talk to you about how animal models influence the research that we're doing and advancements in brain injury treatments. So when I talk with people about how I conduct animal research, I get a lot of questions. I get questions like, why do we need animal models to advance research and education? What kind of alternatives like computers or cell lines can you use instead of using animals? Can the results from animals translate to humans? You know, humans aren't rats. I get that a lot. What have we learned from animal research? Are the animals used in research and education protected by laws? And specifically in my area, how are animal models used to help brain injury? So we use a lot of animal models in research, and as I showed here, we have researchers even at this facility that are using all sorts of models, such as the zebrafish on the top. There is the mouse over here on the left, a rat, rabbit, pigs, and even non-human primates. So all these are being used for animal research, but we need to use them in order to understand the physiological response of how tissues function after disease and injury, and how we can understand the underlying disease. There are alternatives to animal models. We always start as scientists to initiate our research by looking at computational models and cell models. These are really good screening tools. The cells shown here on the bottom, these are fibroblasts. These are your skin cells. But when you think about growing these skin cells in culture, there's a lot of other cells in your skin, right? You have nerves, You have muscles that help you get goosebumps, right? You have blood vessels. You know that if you pull a hangnail. So all these other cells interact and work as a tissue, and they function together. So when you are doing initial screenings with individual cells, that gives you a good idea of how the cell will respond to injury or disease. But it doesn't give you the complete physiological response that would actually happen within the human body. So we really do need to replicate in animal systems in order to finalize designs and treatments for humans. So can these results be applied to humans? The answer is yes. There's many similarities actually between us and a lot of the animal models that we use. And one example that is used a lot is the immune system. Our immune system is very, very similar to the immune system in a mouse. And so a lot of vaccines and other things have been created in mice that we can use in our body. And so it's actually laws are provided by the Declaration of Helsinki that mandate that we have to test things that are going to be put in the human body in animals first to prove that they're not toxic to us and they're safe for the human body. And I like using this image over here because this is from this website that's listed. It talks about all the animal research that has been conducted for Nobel Prize awardees. So actually they state that 96 out of the 108 Nobel Prize awardees in physiology and medicine were directly dependent on animal research. So it's very important to understand that we need to use these animal models in order to advance medicine. And it has a high impact. So just a couple examples of how we've used animal models to advance medicine. One example is cystic fibrosis. If you're not familiar with that disease, it's a genetic disease. And it's a major killer of young adults. And what happens is there's a protein in the lungs that has a certain channel that's dysfunctional because of the gene is defective. And so we actually share 98% of our DNA with mice. And so we are able to use these animals to detect the mutation, to help discover the physiological pathology behind this disease, and actually test for novel therapies in order to help treat these diseases. Another example is high blood pressure. I'm sure all of you are familiar that if you have high blood pressure, it affects your whole body, right? One of the key examples of this is people who have high blood pressure also have a high risk of kidney disease. So when you think about all the work that needs to be done, you have to look at a whole body system in order to evaluate how a problem in one might affect other organs. And because of animal research, it's led to the development of diuretics and other treatments that have helped with defend the kidneys against disease. The last example is stroke. 15 million people suffer a stroke each year worldwide. And because of animal research, tissue plasminogen activator, TPA, is one of the most advanced treatments that people can use acutely to stop the pathology of a stroke. And this is used clinically, and it's because of the work that we've done in animal models. So the question about, are all these animals protected? Yes, the answer is yes. There's federal and local laws that are in place to make sure that these animals are protected. And all the institutes that use animals have to abide by these regulations. And us as scientists are required to do the 3 R's, which are shown here. We are required to show that there's no replacement that can be used for our animal work to find answer the questions that we're answering. Reduction is the 2nd R. So we have to show statistical analysis that we're using the minimal amount of animals possible to have a high impact and answer that question. And then refinement. We have to show that the animals reduce the stress in the animals and the pain in the animals while we're conducting the research. So as researchers, we have to prove and rationalize and defend all of these 3 R's. And the Animal Welfare Act requires that all institutions that do conduct animal research have a committee that evaluates all of our protocols. And on that committee, and we have one at Virginia Tech, it's called the IACUC, Institution of Animal Care and Use Committee. There are veterinarians on that committee, there are scientists like myself, and there are community members, such as yourself, that are allowed to participate in reviewing these types of protocols to make sure that the animals are being used for improve the quality of life of humans. It's important to note that without these protocols being approved, we can't do our research. They don't let us order the animals. And also, there's no federal funding would be granted until these protocols are approved, so we don't get our funds to conduct the research. So I want to just quickly step into my area of research, which is traumatic brain injury. And my area is kind of focused on injuries that occur in military relevant environments. And so a lot of the research that I do looks at how blast from an IED exposure affects the brain. But I want to just talk about traumatic brain injury in general. At least 1.7 million traumatic brain injuries, or we call them TBIs, occur annually. About a third of all injury-related deaths are occurring because of TBIs. And 75% of TBIs are labeled as mild, which is really important because these are the types of conditions that are not visible. If you look at somebody, you can't see if they have a mild TBI. So being able to diagnose and identify these individuals, it's really important to understand the mild levels of these diseases. And overall, there's about $1 billion spent in the US because of loss of productivity of individuals with traumatic brain injury. So we define TBI as a mechanical force that has an effect on the brain. This can happen from what we call a rapid acceleration, deacceleration, like if you're in an automobile accident and your head moves forward and then you come back. A sports-related concussion, I'm sure you've seen all the pictures of the football people getting hit and they're going really fast and then they're immediately stopped, right? So that's the type of impact-related TBI. There's also impact or penetrating from projectiles. And then what I'm going to talk about today is the TBIs that occur from a blast energy. And all these types of TBIs can lead to temporary, but about 20% of these TBIs lead to permanent impairment in cognitive, physiological, and psychosocial responses. So I'm going to list 3 critical needs that is in research right now that I feel that the clinics our research is providing information to the clinics. The first critical need is that there's no FDA-approved blood biomarker for TBI. So what that means is that if somebody feels that they've had a concussion, they go into the clinics, there's no blood marker that they can draw and say, yes, you for sure have a TBI. So that's a big problem because, as I mentioned, individuals with a mild severity of a TBI are unlikely to have a lot of physical symptoms that you can see. Also, what's important about having a blood biomarker, which would be really great for the clinical use, to evaluate types of treatments. If you can see the stages of recovery somebody is undergoing because of a marker and their blood is, let's say, disappearing or increasing. So that's really important, critical need in the field. A second important critical need is that there's no biomedical imaging technologies that can detect a mild TBI. So if you look at this figure here, stroke, on the right, you can see there's a very large dysfunction or area in the brain that they can actually visualize from an MRI or a CT. And for severe or moderate TBI, you can also see. But for the mild, which is shown there on the left, you can tell there's no real structural damage that an individual can see through the biological tools that we have right now. So we're actually working with individuals here at the VTCRI to try to identify different ways of using those tools, but looking at the neurochemistry, the cellular, the small chemical changes in the brain, instead of the structural damages that you see here. And the last critical need is there's really no effective treatments right now for TBI. Many drugs have been tested to try to prevent or mitigate and decrease tissue damage, which some of them are listed here, but they only work for a very small or limited population. So we really need to find better treatment strategies for those with TBI. In order to do that, animal models are vital. They're vital for the advancement of these gaps. And having the ability to study injury-induced changes allows us to look at, in the animal models, you can look at behavior over time and see how behavior could change. You can look at the physiological response over time, metabolic changes or inflammatory changes over time. And that's really important because, as you know, all of these different assessments involve whole body or systemic function. So in our area, in my lab, we're really focused on prevention, identification, and treatment of those who have been affected from a blast traumatic brain injury. So with blast traumatic brain injury, I'm not sure how many of you are familiar with this, but many years ago I was approached by the Army when I was in a large group called the Center for Injury Biomechanics, and there was a lot of interest in a lot of the veterans that were coming back from combat, and they had signs of concussion, and they were wearing their helmets. So the Army said to us, why? Is it possible that this exposure to a blast, which is the weapon of choice right now for our enemy, could possibly be causing brain injury? So we started studying that, and what has been found and noted on this figure here is that 75% of the injuries coming back from combat are related to an explosive device. And that's very different from historically other world wars. And in the TBI population, again, mild TBI is the majority of people being diagnosed as they're coming back as having symptoms of mild TBI. So it's about $40 billion that is spent annually for treatment for these individuals. So I just want to explain to you a little bit about what a blast is. What we're actually looking at is the overpressure that it comes from the explosion. So if you look here, this is the detonation site where you have explosive material that gets vaporized. There's a large fireball. But if you look on the outside, you can kind of see this little line on the outside. That's actually a pressure wave, or what we call a shock wave, and it's pushing the ambient air out, and that's the actual pressure wave that travels a long distance. So in my lab, what we're focusing on is trying to simulate that wave, that if you're standing at a far distance, you're not standing close to this fireball, you're at a farther distance, that you can be exposed to this wave. And this is kind of an example of what I'm trying to show you. Here you can see an explosion in the background. This is a military personnel in a Humvee, and you'll see a wave come at him. See that? So you can see it jostles him, right? He's not getting thrown out of the Humvee or anything, but it's a pressure wave that he feels is consuming him. Let it run one more time. This is another one a little bit closer, and again, you can kind of see that wave. It comes and it impacts the whole body. So that's what we're trying to simulate, is somebody who's in this type of a distance from an explosion. So there's a lot of different injuries that can be caused by an explosion, but we're focused on what we call the primary blast wave right here. You can get secondary effects from the shrapnel that might come from, if you're close enough to the actual detonation. This is a tertiary, where if you're in the wind, it can pick you up and carry you and drop you, and you might physically hurt yourself that way. Or the quaternary is when you're really close, and so you can get chemical burns or chemical or thermal exposure injuries. Again, my lab is focused just on this primary blast wave for somebody that's further away from the explosion. This is what we're trying to simulate in the lab. This is a pressure wave that comes from an explosion, a very instantaneous rise in pressure. Okay? And then there's a decrease in pressure going into a negative phase. This duration here is less than 1 millisecond. So if you think, just to kind of put that in proportion, a car crash or a head impact during a football game, that's about 100 milliseconds. So this is an order of magnitude shorter. And your tissues actually respond differently depending on how fast they are impacted. So it's really important to note that that's what makes this injury different from, let's say, a football or automobile accident injury. We cannot bring explosive devices onto campus, so we designed these blast simulators. This is in the Center for Injury Biomechanics, and it's about a 15-foot chamber tube. In the back here, there's a driver. We put a membrane here that separates this back driver from the rest of the chamber. We pressurize this chamber by adding compressed air. When the membranes burst, a shock wave will go through the chamber, and this is where our test specimens would be. So this is a window that we can take out, and we can insert the specimen. And then, importantly, there's an end wave eliminator that captures and dissipates that shock wave so it doesn't go out into the test lab. And we can produce these really nice, pretty pressure curves. So this is an image of an animal within the blast tube. You can see this is that window that we were showing you. I just want to kind of show you, this is the sling. They're in like a mesh sling. And the sling is tied on the top here and on the top here, and one goes on the bottom. And when the blast wave comes down, they're really just jostled, kind of like that video I showed you. They don't swing and hit anything on the walls. It's just the pressure wave that they're exposed to. So the first question that we wanted to ask is, does this pressure wave cause any problems in the brain? in the brain cells. And what we did is we were able to collect the brains at different times after animals were exposed to a blast. And you can see here we can do brain slices, and then we can use these slices to stain them and look for different markers of injury in the brain. Or we can actually take punches, what we call punches of specific brain regions. Because as you know, the certain regions of the brain will affect different or cause different behavioral problems. So we can try to focus our analysis on a specific area of the brain. And what we found, and this is encompassing years of research, is that there's a significant change of neurochemicals, which is shown here. This is a chemical chromatography recording. You can see there's neurochemicals that are significantly changed, or you'll just take my word for it, because I'm sure that I didn't bring the actual statistics on that. But also, we found histologically, there's a lot of oxidative stress in the brain. So many areas of the brain undergo oxidative stress, and that's something that you guys probably have heard about antioxidants, and then you have oxidative stress, and usually in the brain or in healthy tissue, there's a balance. Well, here there's imbalance. So the oxidative stress is occurring in the cell, and then another word that you guys probably recognize is inflammation. We find that there is a persistent low-level inflammation going on within the brain. But just because you have some of these markers in the brain doesn't always mean that there's a dysfunction, the behavior problems. So we needed to move further and ask, is the pathology or the biological response correlated with the behavior? So we can do that in a lot of different ways in assessments in animals. And one way, looking at memory, is called a novel object recognition test. And how it works is you can place a rodent in an arena, and you put in two objects. And they like to explore. If they're healthy animals, they'll explore and look at the different objects. You take them out after a certain amount of time. You switch out one of the objects, so one of them is novel. And then you put the animal back in. And again, if they're healthy, they'll recognize that that new object is there, and they'll explore that more than the old object. So we do video analysis of all the animals, and we can quantify how much time the animals spend with each object. We also do some anxiety types of behavior assessments. One of them is just an open field. We place an animal in an open field, and again, we're videoing this, and we watch their behavior. And normal animals will explore. They'll go all over the arena. And animals that might be injured will have anxiety-like behaviors, which means they'll kind of hang out along the sides. They don't want to go out and explore. This one is called the light dark box. It's also for anxiety. It's again an arena, but this side of the arena is boxed in, and there's a little shuttle that the animal can go back and forth from the open to the dark side. And my students used to have a lot of fun, probably a decade ago when Star Wars was really cool, and they say, how long does it take the animal to go into the dark side? So obviously if the animal is more anxious, they'll hide out and they want to stay in the dark side versus come out in the open area. So again, we can quantify all this and tell a lot about the behavior that they're showing. So this gives you one result that we got here. This is our control animal. It's called sham. And this is the first test. Remember, they're seeing two objects. And then we take them out and we switch out one object here. And you can see that the animal, when it's red, these are heat maps. So when it's red, that means the animal is spending more time in that area. You can see the normal animal, or control animal, spends the most time with the new object. Compared to the blasted animal, again, they look at both objects here, and then after the wait time, you put them in for trial 2, and again, they don't discriminate with the new object and the old object. So this is considered to have memory impairment. And then with the anxiety, the open field, you can see, as I said, normal animal likes to scatter, go all over, explore all different things. And the animal that's been undergo blast exposure stays along the side and sometimes just huddles in the corner. They don't like to go out and explore. They're a little anxious. So just as a summary, obviously I can't show you all of our research, but I'm trying to give you an idea that we have some primary damage going on. This blast energy is causing some physical change in the brain. That leads to some secondary effects. So the pathology continues with inflammation or oxidative stress. And then that all leads to different memory or anxiety or behavioral problems. So really, using these animal models has allowed us to understand the mechanisms of injury And by understanding the mechanisms of injury, we can then go through and start developing some novel therapeutic targets that might be more appropriate to be used in the clinics, or rehabilitation strategies that might be more appropriate to use in the clinics. So overall, I hope you see that experimental models for TBI are a vital component because it's a very complex injury mechanism, and it will help with advancing in the treatments of individuals with TBI. And some of our future models that we're looking at developing are looking at repeated blasts. So individuals, I have an appointment at the Salem VA here, and I talk to a lot of veterans, and they tell me that within their 1 year tour duty, they might have been, if they're in the combat line, they could have been exposed to 20 or 30 blasts in that 1 year. So we're really trying to start modeling some of the more severe cases of blast TBI, as well as stress. We all know that stress affects us, right? It affects our whole body. It affects the brain too. So all these individuals, when they know they're going overseas, that's a stressful event. They're already in this type of stress environment. They have to say goodbye to their loved ones, and they don't know if they're going to make it back. So kind of modifying our animal models in order to incorporate these other, what I call triggers or additional stresses, might also lead to very valuable information for the future. And gender comparisons. We know that males versus females, their recovery patterns are different. So that's something that we'll be looking at as well. So I think I'm on time. I wanted to definitely acknowledge, this is my awesome lab group here. You can see a lot of young, brilliant undergrads, graduates, postdocs, and research faculty that all helped me conduct this research. Also wanted to acknowledge the Center for Injury Biomechanics and our funds that come from Department of Veterans Affairs. We get funds from the DoD as well as the Office of Naval Research. So thank you. I'm gonna hand it over to you.